Okay, good afternoon everyone. Matthew Cook's my name. I'm Professor of Medicine at ANU and with my colleague Carola Vinwesa co-direct the Centre for Personalised Immunology. And we're going to hear two of our other colleagues from the Centre talking this afternoon. Let me first just acknowledge the custodians of the land, the Ngunnawal people who are gathered today. Let me welcome you all to this special edition of seminar series on Rare Disease Day. I think that this is a very important initiative, Rare Disease Day, as well as the increasing recognition of rare diseases. I think in Australia we're fortunate. We have a comprehensive health care system. Nevertheless, people with rare diseases still confront some obstacles when they're trying to get access to either diagnoses or treatment. And the efforts of Rare Voices Australia and initiatives such as Rare Diseases Day have gone some way towards overcoming some of those obstacles. There's still a long way to go. You know, this operates on a number of levels. We need to increase awareness of the importance of rare diseases, and often that's done by talking about them collectively. There are so many rare diseases that collectively this counts for a significant burden of disease. We need to start to convince legislators of why dealing with rare diseases requires a different approach to some of the ways in which we deal with conventional illnesses. And such an initiative has been introduced and has been championed by Jenny Tyrrell with the SCID screening program that some of you might have heard about and even signed the petition for. So rare diseases are a different, require a different approach because they hinge on the recognition of the fundamental differences in disease mechanism between different sorts of patients. We spent a lot of time in the second part of the 20th century trying to group people together to decide whether treatments would work or not. As we approach rare diseases, we're trying to undo some of that to understand mechanisms of disease. And people with rare diseases are often very generous, both with their time and with their willingness to provide samples, because we recognise here, and you'll hear about this from my colleagues in a minute, that what we understand about the mechanisms of rare disease often have great relevance to what we would consider more common disease. So they give us an important window into mechanisms of disease. All right, so we're fortunate this evening to have two excellent speakers. I'll introduce them both to you now. The first is Dr. Julia Elyard. She's a project leader at the Centre for Personalised Immunology here at the school, and she works on unravelling the genetic basis, particularly of autoimmune diseases such as lupus, but also some of the organ-specific autoimmune diseases. Now, she completed her PhD here at the ANU, won a prize for her PhD, the prestigious Dewar Milne Prize for Immunology in 2008, and then she spent a couple of years overseas at Cambridge and has now returned and works with Carola on autoimmune diseases. And in 2014 she received the Judith Whitworth Fellowship for Gender Equity in Science. David Fulcher is a specialist clinical immunologist and is also a chief investigator here at the Centre for Personalised Immunology. And his research is also targeted at understanding the genetic basis of autoimmune diseases such as lupus and also primary immune deficiency diseases, particularly primary antibody deficiency diseases. He's also a renowned educator and teaches medical students, trainee doctors, scientists, and currently coordinates the Advanced and Applied Immunology undergraduate course here at ANU. So I think they're going to provide us with a joint session now, and so I'd like to welcome both Julia and David to the podium.
Well, thanks very much, Matt. It's a pleasure to talk about this subject and talk about some of the research we're doing at the CPI to address rare diseases. To understand what we're going to talk about, I think we need to focus on the immune system, and most people will appreciate that the immune system is there to provide a response and to prevent us from getting invaded by the microorganisms that we live in. But when we come to study it, we realise that the immune response is not so much an anti-foreign, but a highly intricate system where it also has to not react to self-antigens. And so when things go wrong, disease can result. So under normal circumstances, this is an angry lymphocyte, sort of what I look like on a bad day, but that's attacking the foreign invaders and, while at the same time ignoring self-antigens. But in autoimmunity, the immune system gets it wrong, and that one simple statement is the focus of an enormous amount of research to try and understand exactly what goes wrong in each disease. Whereas in immune deficiency, the immune system fails to respond appropriately and ironically also decides to attack self. Again, areas where we're just beginning to understand it based on our study of the genetics and function of the immune system. So to talk about, with the emphasis on rare diseases and research, I wanted to present two cases of the same condition which I've come across in the 25 years that I've been practicing as a specialist in immunology. And if we wind back to 2008, this is an unfortunate young girl, Aboriginal, rather socially disadvantaged, very difficult home situation, who presented from a very early age with severe infection, including candida involving the mouth, or thrush involving the mouth, esophagus, skin, nails. She had very bad scabies, a condition called Norwegian scabies, where there's actually invasion of the tissues. That should say shingles, not singles. And with the result of chronic disease and malnutrition, we had a great deal of problems keeping Sarah in follow-up, getting her to take her medication with the inevitable effect of multi-resistance from the organisms that she battled with on a day-to-day basis. And unfortunately, she died in her early 20s of the complications of these infections. So after her death, we were able to store some DNA. And when we went to a conference, International Conference on Immune Deficiency, there was a lot of excitement about the discovery of the protein called STAT1, which is an essential regulator of how some lymphocytes do their stuff. And we were able to go back and sequence Sarah's gene and show that the STAT1 mutation was affected in her and responsible for at least part of her phenotype, probably a lot of her phenotype, although she had a very severe course. So fast forward another 10 years to the second patient that I saw with this condition. She had a much milder course than Sarah. She had the same condition in that she had chronic thrush infection of various sites of her body. She incidentally had underactive thyroid, which we thought nothing of, iron deficiency, and she had, I don't know whether these pedigrees mean much, but this is Lucy here. She had a mother who was also affected, 2 aunts who were affected, some cousins who were affected, and her daughter was also affected with a very clear genetic component. So unlike when Sarah first presented, the development of genome sequencing has taken off enormously, and we're able to send her DNA almost immediately for what we call exome sequencing, which is where we sequence all the genes in the body that actually make functioning proteins. And after about a month's wait, the answer came back that this patient also had a mutation in the protein STAT1, explaining her condition. So what does that mean for patients with this rare disease that we know now that we didn't know 10 years ago? Well, a group of investigators had put together over 100 patients who had volunteered their samples and their clinical information to that central research body and was able to construct a very careful snapshot of what patients with STAT1 mutation might expect. It was recommended on the basis of this review that patients be screened for cerebral aneurysm, which is almost an unexpected association with this condition. And that was negative, but in the small print was a higher rate of esophageal cancer, and unfortunately this patient had that complication within months, or was discovered to have that complication within months of the genetic diagnosis. So what else does it mean? Not only do we have a better understanding now of this condition, but we can think of ways of treating those patients based on our understanding of what those proteins do in the body. So this is a signaling protein within the cell, which is overactive, and so scientists will think, well, what can we do to block that overactivity? Are there drugs out there which can do it? And there are actually a lot of drugs which are marketed to treat blood disorders, which are actually very useful in treating some immune deficiencies. So this is one of the reports of the drug ruxolitinib, which has been used. It's actually available in Australia to prescribe, where a first patient treated with this did very well. Subsequent patients may not have done so well, but the principle is that if we can understand the defect in how the immune system is working, we could think of ways to intervene to make that rare disease more tolerable and to try to prevent complications like Sarah went through. So now we have the traditional second half of the 20th century type routine diagnostic test where a patient with a condition goes to a doctor who identifies that it may relate to a genetic syndrome. But now we are able to sequence the DNA for not too much money, $1,000 or $2,000, identify the variants, establish a more specific diagnosis for that particular rare condition, and then possibly optimize the treatment based on what we understand as being the physiology of that condition. So that sounds very good for this particular protein. It's a STAT1, but we are scratching the surface at understanding the role of genes in causing disease. So we are now at the stage where we can find genetic changes quite easily, but we are stuck with the idea of whether those genetic changes that we pick actually cause disease, and if they do, how do they cause disease? And in the case of the immune system, what function is altered by that particular disease? And finally, when we understand that, how can we intervene with drugs that are possibly already available? So we wanted to take this opportunity, both Julia and myself, to talk about what we particularly do at the CPI in addressing similar conditions to this. And this is what I do. The hypothesis is that if you have a genetic change, this will affect the cells that make up the immune system, and that by measuring those cells carefully, using donated patient samples in combination with their genetic code, can we understand more about what those mutations are actually doing. So we need patients with rare diseases. We also need normals, so that we can take those samples, look at the blood cells, and correlate that with what's going on in DNA. Well, we have a lot of blood cells in the body, and these are the major classes, but by using a technique called flow cytometry, where we measure lots of proteins on the surface of those cells, we get a much more detailed snapshot of what the immune system is doing at any one point in time. So, of the humble lymphocyte, we have about 30 or 40 different types of lymphocyte in the body, all working together to coordinate the immune response and to prevent reactions against self. And similarly, with the so-called monocytes, we have different populations of those. So what I set out to do is to measure all those populations independently and form a snapshot in any given patient of changes within those cells that might reflect their disease. So we now have about 300 sample or patient samples where this has been done. We have 70 normal people, we have 230 patients approximately with either immune deficiency or lupus or sometimes autoinflammatory disorders. We throw these through a flow cytometer which measures those subpopulations very carefully, and then we try to depict any variation from normal using a heat map. That basically means in a given patient, how much does each cell population vary from normal? Is it overrepresented, which would be here in red, or is it much lower, deficient if you like, depicted in blue? So this is a typical snapshot of what a patient with immune deficiency might look like, and you can see all the subpopulations of blood cells lined up, and each one with a colour representing how far that population deviates from normal. Now, by doing this to the whole patient cohort, we can then analyse similarities between that snapshot and those of other patients, some of whom will have a genetic diagnosis. And this computer algorithm placed this patient next to a patient with a mutation in a protein called NF-kappa B2, and indeed they share that same mutation, giving some credence to this type of approach. So how else do we help understand how genetics and cellular changes operate? Well, this is a particular group of patients with a disease, and we've sequenced their genome, and we found 2 patients with a similar type of genetic change, and we've done their cell snapshot and found that these particular irregularities associate with that particular gene mutation. So this is an example of a patient of mine. I won't go through the details of it, but I've looked after all of these patients and still do so now, all of whom had various severity of lupus, and a lot of this work has been done by Catherine Bassett, but you can see that our genetic change at the top here in the 3 lupus patients is compared to the other members of the family, And we look for differences between patients who have a genetic mutation versus patients who do not have the genetic mutation, and we try to infer from that whether that particular gene might be having some functional effect on how the immune system is balanced. So I will hand over to Julia in about 30 seconds, but just to summarise, we take our patient samples, take the DNA, compare it with the cell snapshot, and use those two in collaboration to try to predict known DNA changes based on the cellular composition, and also to look for new gene changes that may alter that snapshot uniquely. So Julia will take this on to talk about how we understand or how we can analyse the function of any specific genetic change.
Thanks, David. Okay, thank you. So in the second half of the talk, I'm going to take you through the process that we use in the research laboratory here at JCSMR as part of the Centre for Personalised Immunology to take that genetic information and look at a gene and actually prove that it is the causation of the disease. So what I'm going to talk about is really the research for new causes of disease. So as David alluded to, this model for the Centre for Personalised Immunology is that we start with a patient and hopefully complete this circle to end with an altered or improved treatment strategy for the patient. And in doing that, we perform the phenotyping of the patient's cells, which David has just introduced, the immune phenotyping of the cells, and we also perform whole exome or whole genome sequencing to take a look at all of the genes within the patient and identify changes from what we think are normal. Okay, so, but that's quite, it's a very easy statement to make, but the reality is that each one of us actually contains many different changes in our genome, and most of those are not causing disease, and they're actually very benign in nature. They might have small effects, but really they have no impact on our lives at all. So how do we differentiate those genes that do cause disease from those that perhaps don't, those gene changes? So we have a filtering process which we use to come up with candidate genes. So I often think that we're acting a little bit like gene detectives. So we have our list of suspects, so they are our genes. But we need to prove, we need some sort of proof. If I was a real detective, I couldn't just line up my suspects and say, well, I think it's you because you look the most like you have committed this crime or you have caused this damage. We need to have some sort of proof there to actually show that it is the cause of disease. And so to do this, we need evidence. And what is the evidence that we use in the lab? Well, it's the samples that we can get from the patients. So it is our patient cells, as well as cell lines or mouse models which we can create to mimic what we see in our patients or the gene changes that we see in our patients and see if they also develop disease. So why do we need patient samples? Well, firstly, the samples that we are talking about in immunology and we regularly get as part of the Centre for Personalised Immunology really are DNA as well as blood cells. And we can use these, as David has alluded to already, to give a diagnosis. So this might be a gene change that is already known to cause a particular disease, and that might change the treatment that is given to that patient. However, in some cases, the genes that we think might be causing the disease have never been linked to that disease before, and we need to understand how they're causing their disease. And so that involves a lot of research in the lab, and then hopefully that can give a diagnosis to that patient, and that may then impact on the treatment. Alternatively, understanding the causes of disease can also, and using the models that we have, might also reveal new treatments that might be used down the line for those patients and that have or have changes in those particular genes. So I'm going to tell you tell you today about sort of each of those cases. So the first one I'm going to talk about is just a diagnostic case. So this patient is Josh, and he is a 16-year-old male. He suffers from CMC, which David introduced before, candidiasis, as well as having an underactive thyroid, vitamin D deficiency, and a delayed onset of puberty. And so the whole exome sequencing of Josh showed, and his genome showed that he had a novel, so that is a new undescribed mutation in STAT1. So this, we decided, was probably the most probable cause of his disease, but he also had a number of other changes in other genes, which also we thought could be contributing to the disease. So because this particular gene change had not been shown before, we wanted to get some evidence that actually this probably is the most probable cause of his disease. So to do this, we could compare his immune cell phenotype to that of another known STAT1 patient. So this is patient Susan, who David introduced before. And you can see here that Josh and Susan share a couple of key cell parameters here, which says that they look very similar. Okay, we were also able to take Josh's cells and activate them in the lab and look at the levels of STAT1 protein. So it's known that these mutations, in STAT1 that cause particularly CMC often result in a hyperactive protein. And here you can see that Josh is in the red here, that he has more of this activated STAT1 protein compared to our healthy controls here in the black. So this is quite nice. We were able to take our candidate gene and actually show that, yes, this is indeed a gain of function variant in STAT1, and it's probably the most likely cause of his disease. Okay, so moving on, the other cases that I'm going to tell you about today are patients that have systemic lupus erythematosus, or SLE, as David also briefly introduced. So before going on, I just want to tell you a few things about SLE, which is the disease that we work a lot on in the lab. So SLE is an autoimmune disease, and it affects mainly women, with a 9-to-1 ratio of affected women to men. And it's a very complicated disease. So patients with SLE can have any one of a lot of different disease manifestations and symptoms. And in actual fact, we probably think that rather than being one disease, the causes of SLE could be different in different patient groups, and it's really a clumping of diseases together that may have quite different underlying causes and perhaps mechanisms. However, there are a few unifying factors. So a lot of the disease in lupus, so I'll just refer to SLE interchangeably with lupus, is driven by autoantibodies that can be found in the blood. And these autoantibodies can join together to form immune complexes, which then trigger the immune system. They send out danger signals to induce inflammation through the production of a particular types of molecules known as cytokines. And particular interest in SLE, and the ones that I'm going to talk about a little bit later on, are interferon, so particularly interferons alpha and beta, as well as a molecule called IL-6. So the case I'm going to tell you about, where understanding the cause can perhaps open up avenues for different treatments of a patient is the case of Katie. So Katie had really severe lupus from 2 years of age, which is very early onset to have this disease. And when we looked at her genome, we found that she had a novel gene change in a particular gene called TREX1. So this is Katie here. She's the youngest of 5 siblings and the only affected member of her family. And here is her DNA. So this is DNA sequencing here. And you can see that in this particular position here, Katie only has one allele, so that's the A allele, whereas her parents each have one A and one G. So she's inherited one A from each of her parents. And having this A here in the gene results in a change in the protein from an arginine to a histidine of the protein. And when we looked into her family a little bit more, we saw that Katie was the only member of her family who had two A copies in the TREX1 gene, whereas everybody else had one A and one G. So what does TREX1 do? Well, this is a cell, and normally in a cell we have a cytoplasm and a nucleus, and DNA only exists in the nucleus of the cell. However, if your cell was to be infected by a virus, what happens when that virus replicates in that cell is that you get DNA from the virus in the cell cytoplasm. And this is recognised by the immune system as not being correct. We shouldn't really have DNA in the cytoplasm, and it results in a cascade that results in production of these inflammatory molecules I introduced earlier, known as interferon. This is like a big danger signal, and it starts the immune system off to get rid of the virus from the body. Okay. However, even in the absence of a viral infection, occasionally you do get small amounts of DNA in the cytoplasm. And this is just part of normal cellular processes. And the job of TREX1 is to basically chew up those small bits of DNA and stop the inflammatory cascade. However, in the cases of people like Katie, who may not have a functional copy of TREX1, you get build-up of that DNA in the cytoplasm of the cell, and that induces inflammation. Oh, sorry. So what we wanted to know was, indeed, does Katie have a functional TREX1 protein? So we identified a genetic change which we think could be impairing function, but we need some proof. So to do that, firstly, we could look at the TREX1 protein itself. And so here we have normal TREX1, and then this is the mutation or the variant of TREX1 that Katie has. And you can see here, this is this normal TREX1 has been able to chew up this piece of DNA, and you get this lovely ladder showing the different sizes of DNA, whereas Katie's TREX1 was not able to do that. And secondly, we thought we could look for markers of interferon. So it's known that when your body produces large amounts of interferon, that you can have a number of genes that are upregulated. And so here we can see we've got in this heat map a whole heap of genes that when you treat the cells with type 1 interferon, they go up, so they're red. Okay, and so then we look at Katie's cells, and we see that even though we haven't actually treated them with interferon, that they are actually, a lot of these genes are red, suggesting that she has a lot of interferon just floating around in her body. And luckily for us, we also have a mouse that also has a variant in TREX1, which is in a very similar position to Katie. And we found that like Katie, this mouse, or these mice that have this variant in TREX1 also upregulate all these genes and have large amounts of interferons around. And these mice also develop features of lupus-like disease, including autoantibodies, particularly ANA antibodies and anti-double-stranded DNA. And perhaps most interestingly, I told you that this was a case I was going to tell you about treatment. So what this means now for Katie is that there are quite a lot of different therapies out there that target excess type 1 interferons. So, and some of these are available in the clinic in Australia, and some are still undergoing clinical trials. But it means that she is perhaps a candidate to have a different type of therapy and move away from the non-specific therapy which is currently on, or if her disease was to get worse, she could have a different therapy. Okay, so the last thing that I wanted to talk about today is understanding the disease mechanism. So 10 years ago, this was a really big undertaking. But in the last 5 years or so, there have been a development of a particular technique known as CRISPR-Cas9. And this allows scientists to target particular parts of the genome, and they can cut a part out and replace it with something else. So we are now using this technique, we are able to make either mice or cell lines that have the same genetic changes that we see in our patients. And this gives a really powerful tool to understand the mechanisms of disease, particularly in cases where we don't know how the genetic changes that we see might be causing disease. So I'm going to tell you about a couple of mice and some cells that we have that we've made to replicate our patients. And so I just want to first recap that part of the pathogenesis of lupus is that the body produces autoantibodies, and that these can form immune complexes and lead to the production of cytokines, particularly interferons and IL-6. And that the cells that produce these autoantibodies are known as lymphocytes, the B lymphocytes or B cells. And so Simon Jiang, who's another co-investigator and colleague from the Centre for Personalised Immunology, identified that we had a number of patients that had shared a mutation in a particular gene that was found in B cells, and that gene is known, I'm just going to call it gene A today. So what he was able to find was that when, so these patients that had a variant in gene A, they produced more IL-6 compared to healthy controls or some other lupus patients even. And looking at the mice that shared or had the equivalent variant in gene A, they also produced more IL-6 compared to normal litamate or wild-type mice. And interestingly, some of these mice, it's been still sort of figuring this out, and it's been a little bit variable, but some of these mice have developed features of lupus disease, such as autoantibodies and also immune complexes that deposit in the kidney and can lead to impaired kidney function, which is also seen in a lot of lupus patients. However, one other thing that Simon noticed was that some of these, or that one of these patients particularly produced another cytokine, large amounts of another cytokine called, known as IL-10. And interestingly, that patient also had another gene variant in a second gene, we'll just call it gene B, and when in the lab, our colleague Vicky was able to make cells. So these are a cell line, a lab cell line that was induced or engineered using CRISPR-Cas9 technology to have the same variant in gene B that we saw in the patient here. They saw that these cells also, when treated, produced more IL-10, suggesting that gene A might result in more IL-6 in the patient, and the variant in gene B might be responsible for increased IL-10. And we're still working out how these are contributing to the disease. But I just wanted to show you that it's really important to have the patient samples to compare to, because we wouldn't have necessarily known to go back and look for another gene without the patient samples. So in summary, what David and I have talked about today is that when we have a new genetic change is identified, that perhaps research can use patient samples to prove a change in the gene or protein that changes the function, infer immune effects by changes by taking a cell snapshot. Oops. Create mice or cell lines with the same gene change as seen in the patients, and to compare features of the patient samples and mice, and to use these to understand the disease mechanisms. We can then perhaps choose therapeutics based on the ability to reverse specific immune defects. And I haven't really talked about it yet because we don't really have the evidence yet, although we're working on it, to use these mice or cell lines to test therapies that might be better targeted to that particular cause of disease. But all of this is really dependent on having samples from the patient, and without that, we wouldn't be able to do this work. So I'd just like to finish with acknowledging there was a lot of people who've contributed to this, and I'm sure I've left people off, so I'll just apologise now. But other members and CIs and executive team of the CPI, other members of the lab, as well as services at the John Curtin, and in spirit of today, a big thank you also to the patients and their families, because without them donating their time to give samples and the samples, we wouldn't be able to do this work.
Well, thanks very much to Julia and David. I think we've got some time for some questions, if anyone has some. Paul.
One of the things that you suggested may be common to the various forms of SLE were the elevated levels of interferon, for example. Are you yet at the stage where you can say that all SLE is due to, it's a common property of all of the multiple different forms, and all the genes do, all the different variants do, is increase the amount of interferon that's being produced? And the second thing is, how effective are the antibodies to interferon? Clinically, you've got all those options available to you. Are they effective? We know that they're very expensive, so they have to be pretty effective to make them worthwhile.
Yeah, so I'm going to let David answer that. I can give that answer to this. So maybe the first part, in terms of the research that has gone on, I think there's a suggestion that anywhere between sort of 50 to 80% of patients, lupus patients, have this elevated interferon signature, and that interferons might be really important in driving their disease. But there are some patients who don't have it, so I think there probably is a subset of SLE that isn't dependent on interferon. But then also, I think whether that interferon is the cause and the driver of disease or an exacerbator of disease isn't probably clear yet.
And in terms of the treatment?
Yeah. Well, I think the interferon also has been found in other rheumatic conditions. It doesn't seem unique to lupus, so I think there probably are other factors involved. And 2 trials have been done, one of anti-interferon alpha and one of the receptor for interferon alpha. I think the former one, I always get them mixed up, but one of them showed no benefit and the other showed pretty good benefit. But interestingly, from my recollection, the stratification based on those with a high interferon release didn't actually show what was predicted, that if you had a high interferon, you didn't necessarily respond. But they were pretty effective, and they're yet to be released in Australia, but these initial studies were good.
So this raises an interesting point about how we might approach stratification of patients in future for clinical trials. We all, one of the hallmarks of clinical trials in lupus is their failure compared with many other autoimmune diseases. It's been very hard to get a convincing effect, and this is a really important point that just taking all comers and trying to evaluate them with new drugs has been disappointing. So maybe you might like to comment on how we could rethink our approach to getting some of these novel agents into the clinic in the light of the sort of work that's going on.
Yes, well, I was going to get up on my soapbox for a minute, and I think that introduces it well, and then I might talk to that, if you would indulge me. But I thought on Rare Diseases Day, even though this is a little bit off target, I would talk about 2 or 3 of my patients that really annoy me. Not that they annoy me personally, but the failure to be able to treat them appropriately really annoys me. So this is one patient that I see. He's got adult onset Still's disease. So unfortunately, that's not rheumatoid arthritis. It looks very like rheumatoid arthritis, but it just isn't. He's had severe disease for most of the last decade. He's been on steroids, methotrexate, mycophenolate. He's been on all 3 of those in combination. He's recently been on cyclosporin, but the best treatment for him is one of these 3 biologics, and I've put the price on them per month, but he doesn't qualify, so he can't get access to it, even if he's stratified to show that that was useful. And part of the well, I'll present the next case. This is the other guy. He's got a rare condition too. He's got what I think is Schnitzler's syndrome, although there's no diagnostic test, but he's got, his life's a misery. He has continual fevers, tiredness, all his lymph nodes and spleen are enlarged. He gets daily itchy hives, which are resistant to standard measures. His inflammatory markers are always high, and he qualifies, well, he would, the best treatment from looking at worldwide cohorts of this condition is anakinra. He can't afford $500 a month. That's more than he's paying in rent. And anakinra is a drug that's on the market for one reason only, and interestingly, it's another rare disease called CAPS, or cryopyrin-associated periodic syndrome, but he doesn't have CAPS, so he doesn't get the drug. The third patient is the one I presented earlier with a STAT1 problem, and ruxolitinib is $5,000 a month. So, so this frustrates me, because if you have a patient in front of you who you know needs a biologic of these types, drug companies don't tend to market to rare diseases. They market to common diseases. So all the drugs that I've mentioned, or 2 of the drugs I've mentioned, are marketed for rheumatoid arthritis because they're common. But these rare diseases, they're not marketing. Now, part of the reason, you might say, well, that's a bit unfair, and it is unfair. But part of the reason is what Matt was alluding to, that these diseases are so rare that you can't demonstrate in a randomized fashion that they are effective for a rare condition. And so, the future, I think, is to try and relax some of these requirements for randomized trials, particularly in the era of personalised medicine, where we can come up with clear immunoregulatory problems that we know we have the drugs to target, and we may need to rethink the way that we use these drugs and how we get them funded. So at the moment, self-funding these things, unless you've got lots of money, is not unrealistic. For me as a community practitioner, I can't get hold of these drugs. I rely on cooperation with people like Matt and other colleagues to get these drugs funded through the public hospital. But I've included on the screen this message that I sent to one of my colleagues in Sydney about the possibility of getting anakinra, and that was the response. So even in the public health system, in the public hospital, they will not fund it, even though it's well regarded as the most effective drug for this rare disease. So I think if I have to say one thing regarding rare diseases or the rare voices, we need a mechanism that bypasses this. We need to rethink the way that these drugs are funded. It shouldn't depend on where you live. It shouldn't depend on who's holding the budget strings. It should be open and equitable and fair in order to get these drugs that are marketed for common conditions but banned for patients who happen to be unfortunate enough to have a rare condition.
Thank you. Another question.
When talking about funding of these drugs, could they be funded as part of the research project?
Well, research funding is even worse. So ask a researcher how they get their money, and it is a continual battle for funding, both to do the project and indeed for their own salary. So once you've got some money for funding, the last thing you want to do is blow a quarter of your budget on monoclonals. And in general, the NHMRC tends to frown a little bit on spending money on therapeutics.
But you're talking about rare diseases, so it wouldn't be a very commonly occurring expense. It would be something that is fairly uncommon.
I agree. And I think if you add up all the rare diseases together, they're not uncommon. But any one of them is rare.
Yes, so I think that this is a really important question. NHMRC currently does fund some conventional clinical trials where large groups of people are put together and given a drug, and we try and work out which one works. But this is part of the required change in mindset that David's referring to, because there are sorts of clinical trials that you can do which involve very small numbers of individuals, sometimes only one individual, which is what we require in this sort of situation. So it's a different method that needs to be employed, but it's also a different mindset that's required to recognise that this is a worthwhile endeavour, either as a research initiative or as a means to gather the evidence to change the way patients can actually get access to drugs.
But is it really only a very small number of individuals? And you mentioned new causes or newly identified causes. So could that actually mean that there are patients that the cause hasn't been identified?
Absolutely.
It may apply to
It may well apply to a lot, and I think that this is the other component of this. If we think about conventional clinical trials at the moment, where we're applying these sort of precision therapies, then you can understand from what David and Julia have already outlined that there's only going to be, we would only expect that there will be a small proportion of those individuals who are recruited into those trials who are going to respond to that very targeted therapy. And we're not gathering that information at the moment. So that's another reason that approach is is no longer valid. It's a bit of an anachronism to be using that strategy for evaluating these precision therapies. All right, well, look, I think it's been a very stimulating evening. I thank again Julia and David, so join with me in thanking you. Thank you for your interest, and I think we have some refreshments outside for those who are interested.
I'm going to get you wrong. You don't want to be the person who uses the colon, the week before it's registered on the PBS. I think the other thing I would suggest is getting your patients to write to the local members and putting the pressure on the administration politically.
Well, I have enormous trouble.
I'm with you.
And it shouldn't be geographical.
It shouldn't be.
I remember this one case I wanted to put on TNF blockers. I can't remember what they had.
Yeah.
They said, yes, we'll fund you, but for 3 months.
Yeah.
And what is the point of that?
But then you show that it works. And
then you show that it works.
But that's
what
what
this works. You can't worry about it.
So I don't share your view. I mean, I have a lot of, I spend half my time now writing special prescriptions to get patients on drugs from various sources. But in the DTC, I find refractory, and I find the whole setup is wrong because, they put the acid on the local clinician, whereas they should be aggregating the data and taking it upwards and saying, right, let's see what everyone in Australia is doing so that we can gather the information.
If you had a centralised system, you'd say, yes, I'll give you that drug.
Well, that's for Australia. Yes, the DTCs are meant to all respond to the central, what's it called, INTAG or something. They're all meant to feed into that. They don't.
I'm just
Yeah.
enjoy the talk. Yeah.